All right, so we're naming this particular molecule. All right, so when you think you have it, let's see it in the chat. Let's see what we all remember where we left off here. Cool. All right. So remember, we have the same standard procedure anytime we're naming something. First of all, you got to make sure that all of those carbons are accounted for in your name. That's the goal, right? So in this case, this cyclohexane ring would be considered like our parent chain. And it is not just hexane, but cyclohexane. I have these two substituents. They are both methyl groups. And they are located 1, 2 carbons apart. So this would be 1, 2 dimethyl, methyl cyclohexane. But one of the other things that we learned about was cyclohexane, and that's what's represented with these wedges and these dashed bonds. Remember, a wedged bond like the one on the top on carbon 1 indicates that that bond is pointing out at you, and the dashed bond is pointing into the page, right? So it's sort of these different representations. Since they are pointed in opposite directions, this would be considered the trans isomer. And so this would be the full name with the stereochemistry included. Okay, and if I were to draw the other stereoisomer, I would now just make the bonds either both wedges or both dashes. Both would be considered the same stereoisomer here. But so this would be cis 1, 2 dimethyl cyclohexane. And again, this is a pair of stereoisomers. Okay, so now we're going to sort of work on marrying this sort of idea of these bonds pointing in different directions with this conformation of cyclohexane here. Before I do that, I actually found, let me just show you real quick. Okay, this website here will just sort of let you see the conformation of cyclohexane more clearly. I put it in, if we go home, we can use this hot button here, or if you're on the mobile version, you would go to modules, which I need to get rid of some of the stuff. It's a busy menu. Scroll down to this chapter and go to what would be today's lecture. This was where today's lecture video will be posted. I posted a link to this website. So if you guys want to take a second to go there so you can follow along with what I'm doing. But basically the whole idea is it'll let you take this model of cyclohexane and spin it around to get like a little bit more familiar with it, because I know that picture that I just showed you of all those axial bonds and equatorial bonds is not necessarily the clearest thing. It helps when you can kind of spin these things around. Right, so here's the cyclohexane that we're used to, looking at as, you know, the hexagon. And so if we flip it on its side, we can start to see what that model was telling us, right? So again, we have 3 carbons, or every other carbon is pointed up, and then every other carbon sits lower down. We have on these pointed up carbons here, you can see you have these hydrogens sticking straight up in the air. These are what we would call the axial position on these carbons. We have axial positions on the other carbons too, but they're pointed straight downward. And then each carbon also has an equatorial position, which is sticking sort of straight out from that ring. Okay. So if we look back, so in this case, they're all hydrogens, but we can imagine starting to replace these axial positions or equatorial positions with these methyl groups that we saw on our 1,2 dimethyl cyclohexanes. And so you'll note that if we have a trans 1,2 dimethyl cyclohexane, then both of those groups would be in the axial position, for example, because on one carbon, they were on one side, so pointed up at you. And on the other carbon, it was pointed into the board. Okay, so this is sort of what I mean by we're going to marry these two concepts that we learned. But again, go to that website and take this thing and spin it around. It just kind of helps to get a sense more of, you know, these different positions and what they mean and all that good stuff. Again, importantly, every carbon has an axial position that's either pointed straight up or straight down. And they also have an equatorial position, which is pointed straight out from the ring. I mean, importantly, this isn't something that you would like know, just, you know, you shouldn't be like, oh yeah, of course, duh, I knew that. No, I mean, this took some research to figure out what the exact structure of cyclohexane was, a lot of modeling, but this is, you know, this is it. This is what the data has said, right? The structure has these different positions here. Okay, so again, if we take this kind of image, let's see if we can copy this. No, okay. All right, so we have this idea of our trans structure. So now we can imagine, let's say, right, so this is carbon 1 and 2. I'm going to kind of arbitrarily pick those 2 to work with. You could have chosen any pair of carbons that are next to one another and sort of done this. Here's carbons 1 and 2. So if we have this methyl group right here pointing out of the page, then that would be in this position right here, in this axial position. And then if we have this methyl group right here pointing into the page on carbon 2, that would be this axial position here. Okay. So we can see that in this structure that we've drawn here, these 2 methyl groups are both sitting in the axial position. Okay, but then this structure actually can flip back and forth between between these 2 chair structures. And so what happens if we want to just sort of, actually, I think, well, you can see it on that website that I showed you as well. It will give you the structure for both of them, but just to sort of follow along here, let me get different color codings going on. We'll see carbon 1 was pointed up in the 1 conformation. And then this chair flips, and now carbon 1 is pointed down. Carbon 2 was pointed down, and then it flips, and now it's pointed up. Carbon 3 was up, now it's down. Carbon 4 was up, or was down, Now it's up, et cetera, et cetera. Okay, so the carbons that used to be pointed up are now pointed down. And the carbons that used to be pointed down are now pointed up. All right, so cyclohexane is flipping back and forth between these two. If you have just regular old cyclohexane, no substituents on there, then there's an exactly sort of like 50-50 split between these two conformations. I'll talk a little bit about the boat structure. It's really not anywhere near as consequential as these two here. So I want to spend some time getting familiar with these structures, but I will talk a little bit about the boat. Okay. The other thing that's really, so again, a cyclohexane without any substituents will spend an equal amount of time in both of these conformations. It's actually constantly sort of flipping back and forth and back and forth and back and forth. The other thing to note about what's happening here, and they've kind of color coordinated here, is let's focus on carbon 1 again. Carbon 1 has this red bond that's axial in the 1 conformation. Well, when the structure flips, that same bond is now in the equatorial position, right? Likewise, if we look at carbon 2, we can pick one of those two bonds. Let's look at its equatorial bond in blue here. When carbon 2 flips, all of a sudden that bond becomes in the axial position. Okay, so when this flip occurs, so again, cyclohexane is 50-50 here. When this flip occurs, axial becomes equatorial, and equatorial becomes axial. Okay, so we have cyclohexane. It's kind of flipping back and forth and back and forth. And when it's doing that, it's switching its bonds that were once axial or then equatorial. The equatorial bonds become axial, and then it'll go back again, right? Just constantly kind of moving back and forth between these two conformations. The letters are changed because they represent the quote unquote axial bonds with an A, which are the ones that are going to be pointed up, and the equatorial bonds, which are going to be pointed out. And so what's sort of being consistent, held consistent between the two is the color coding, right? So you can monitor what happens, for example, to the red bond on carbon 4. It goes from being A, axial. Now in the other conformation, that red bond is E, equatorial. Okay. I'm not going to expect you to be able to draw these chair conformations. And in fact, I'll give you this kind of model to work with to answer these type of problems on an exam. So you can kind of get comfortable with looking at these two structures here and kind of picturing these different groups on top of them. Okay. But for real quick, before we sort of dive into complicating things, let's just all make sure that we understand that cyclohexane isn't sitting as a flat hexagon. It's actually sitting in this, what we call the chair conformation. In that chair conformation, its two bonds have two different positions, one axial and one equatorial. All right. Not only is it sitting in this chair conformation, but it's actually flipping back and forth between these two chair conformations. And what's happening during that flip is axial bonds are being turned into equatorial bonds. Equatorial bonds are being turned into axial bonds. Okay. Before we kind of, so let me take away all my markings, get it naked again. So before we move on and kind of complicate things, does anybody have any questions about what this structure is saying? Again, we can follow a particular carbon because they're numbered. So carbon 1, that I can match that. The particular bonds are colored. So this pink bond on carbon 1 is the same bond as this pink bond on carbon 1 once it's flipped. And then again, the differences between the letters are telling you whether it's an axial or an equatorial. And we can see that the axial bonds, this particular axial bond is becoming an equatorial bond in the flip. Okay, again, this is not something you're going to just like be like, oh yeah, duh, of course it flips. No. You have to be told this sort of thing, right? But this is the reality. This is the conformation of cyclohexane. So any questions on that before we move on? We all semi-comfortable with this, this structure here? Okay, so now again, so this would be cyclohexane all by itself, no substituents. Would be a 50-50 kind of distribution here. If we now look at our dimethyl cyclohexane, again, let's sort of just color our bonds. So on carbon 1, there's one pointing out at us. That would be this one here. And on carbon 2, it's the trans structure. So the other one is pointed down. So that's this bond here. So both of these are in the axial position in this structure, in this conformation. And then the chair is going to flip. Right. So actually, let me just label this one is both axial. If I monitor what happens to those same bonds during the chair flip, now on carbon 1, this red bond becomes equatorial. And on carbon 2, that red bond after the flip becomes equatorial. Right, so on the one, they're both axial, and on the other, they are both equatorial. Okay, so far so good. Does everybody see how I got that? Can I get like a thumbs up or something if we're following along here? All right, so again, and I'll give you this sort of the model that I was just drawing on. So you can do the exact same thing that I did. Go in there and kind of match who's who, which way they're pointing, and see how they're going back and forth in the flip here. The key thing that we're going to note here is that unlike cyclohexane, this will not spend an equal amount of time in both of these two conformations. In fact, 99% of the time, it will be spent in the conformation where both those methyl groups are in the equatorial position. Right, so cyclohexane, they're all hydrogens. Both of those conformations are equivalent. As soon as we make these methyl groups, right, so now this represents a methyl group. This represents a methyl group. I can do the same thing here. So as soon as we make those methyl groups, now all of a sudden that chair conformation is going to reside in the conformation that keeps those two methyl groups in the equatorial position rather than the axial position. Okay, can anybody guess why that might be? Right, why doesn't the chair like those bonds in the axial position?
Is it because the molecules are heavier?
It's not so much heavy, but I think you're getting at this idea. It's actually size, right? Those methyl groups are big and bulky. And so what happens when they're in this, if we like move my 1% over here, what happens when they're in this axial position is they're really close to all the other bonds that are also in the axial position. Right? There's what we would call steric clashes going on. A general rule in chemistry is big groups, big molecules don't like to be next to other big groups. Right? So this conformation where they're both in the axial position is higher energy because we have this big bulky methyl group clashing with all the other axial positions. In this case, they would just be hydrogens in that position, but nonetheless. So big groups don't like axial. That's kind of the underlying principle here. Okay, if they can move out, if they can put themselves in a conformation where, in this case, this was ideal because this chair can just flip and both of those big bulky groups are in the equatorial position. Great, then it's just going to chill there, right? It won't flip back to where they're both in the axial position. Okay. So let's just do a bunch more examples working with these structures here. Because again, this is sort of I'll give you this shell. I'll give you this picture here. And you need to be able to work with it. Okay, and I'll show you what I mean by that here in a second in terms of the types of questions that you'll be asked. But yeah. Okay, so now let's do another one where I'm going to okay. So this would now be cis 1,3 dimethyl cyclohexane. So using our little structure here, I want you to see if you can identify which bonds would be the our methyl groups in the axial or in our structure. Let's call this conformation A and in conformation B. Right? And we can just use their numbering system. We really don't have to, but it's sort of equivalent. All right, these lecture slides are posted. I know I don't work with too many of the actual lecture slides, but it might be a good idea to go ahead and grab this one in particular and have it like so that you can kind of mark it up and you know, sketch on top of it and such. If this were the 2-semester class, we would spend a whole day learning on how to draw these cyclohexane conformations, but we're not going to bother with that. So again, I'll give you this picture. Just got to be able to use it. Okay. So if I'm going to look on my conformation A first, again, in this molecule, both of these methyl groups are, as pictured, are pointed out of the board. So on carbon 1, I'm going to say that this is this first methyl group here. And give that a CH3. Okay, so then on conformation A, where would the methyl group be on carbon 3, in the axial position or the equatorial position? Let's use the chat. Okay. So I could have actually started with either A, you know, conformation A or B here. I kind of just stuck with one of them. But as soon as I put that methyl group in the axial position on structure A, that means that the methyl group on carbon 3 is also in the axial group at that conformation. Right, so this is also a methyl group. Okay, so now take a second and make sure that you can spy where these bonds are when we move over to structure B. All right, so again, to just use my color coding here on carbon 1, this red bond flips and is now in the equatorial position. And same thing on carbon 3. This bond flips. So now my methyl groups are both in the equatorial position. So which conformation is going to be more stable, A or B? All right, remember the guiding principle here is that big groups do not want to be in the axial position, especially having two methyl groups next to each other in the axial position. That's like an absolute no-no because they're both big and bulky groups. So in this case, structure B would be preferable because it keeps both of those bulky methyl groups in the equatorial position. Okay. Let's do another example here. Okay. I want you to take me, do this whole thing where you're going to sort of go on your own and map out where these methyl groups should lie in each structure. And I want you to tell me whether you think the A or B conformation is going to be more favorable for this particular molecule. This would be cis-1,2 dimethyl cyclohexane. All right, so everybody see if we can take a minute to go through the process that we just did, where we're going to map these methyl groups onto particular positions on each one of these conformations, and then determine which conformation we think is the most stable. All right, so first of all, let's use the chat here. For conformation A, did you find that these two methyl groups were both in the axial position, axial, or both in the equatorial position, or was one of them axial and the other equatorial? Okay, so let's go see. So again, what I'm going to do is I'm going to look, this is carbon 1, this is carbon 2. I'm going to say, all right, this methyl group right here in this structure is indicating that it's pointing into the page. So I'm going to go to carbon 1 and I'm going to pick this bottommost bond. Right, the axial group, the one on top would have been the one pointing out of the page. So this one will be the equatorial group. And then on carbon 2, this group right here is pointing, pointing out of the, what did I say? The dash bond is pointing into the page. So that's why I picked that equatorial group. This wedge on carbon 2 is pointing out of the page. So that's going to put it here. No. Oh.
So when we number the cyclohexanes, is it always clockwise?
It doesn't actually matter for in this case. What I did was to use the same numbering here that they used here. And that's how I was able to match those. Okay, so then, so in this case, I messed this up somehow. Anyways, okay, let's roll with it here because this is definitely, I think I might have messed up on a previous example. So we'll just have to do a bunch more. Okay, so then so in this case, they would both be in the equatorial position. So then when I flip my orange bonds here and my green bond is here, both in the axial position. Okay, so then which one of these positions is preferred here, or which one of these conformations is going to be more stable, A or B?
A.
Right, so again, that guiding principle is we want to keep these bulky groups in the equatorial position. So wait, we got to do the trans structure again, because we're going to do trans 1, 2. All right, so now both of these groups on carbon 1 and 2 are in the same, are pointed in the same direction. So this, we'll color this green one on carbon 1 is pointed out. That would be this group here. We have this pink on carbon 2 that's also pointed out. So that means that it's going to be this equatorial bond here. Right? If it was a dash, like we saw on the previous example, that means that it'd be pointed into the page in this axial position, but it's a wedge. So it needs to be the bond that's on the top there. So then when we flip, this bond becomes equatorial, and this bond in pink becomes axial, right? So every time you flip, equatorial becomes axial, axial becomes equatorial. So then which one of these two structures is going to be preferred? Which one of these conformations is more stable, A or B? Everybody take a second to look at this figure that I've just colored in here. Tell me which one of these conformations do you think is going to be more stable? All right, so hopefully everybody can see that they are equivalent, right? In this case, A nor B would be more stable. They are both equivalent because one has a methyl group. Structure A has a methyl group in the axial position and a methyl group in the equatorial position. And structure B has a methyl group in the equatorial position and in the axial position. All right, so neither one of these conformations is going to be more stable than the other. So in this case, there would be this 50-50 split. Okay, let's just do a bunch more here because I know this is kind of weird. All right, we got to go through and match and then figure out which conformation is going to be more stable. So now we'll do the trans structure for 1, 2, 3, 4, 1,4-dimethyl cyclohexane. So you're going to be looking on carbon 1 and on carbon 4. All right, so I'm going to say on carbon 1, since that bond is pointed out of the page, that it represents this axial group right here. So then on carbon 4 in conformation A, is that methyl group in the axial position or the equatorial position? You got 50-50 shot of getting it right. Put down, let's put down what we think. All right, so in this case, this bond is pointed into the page. Well, that's not a different enough color. This bond is pointed into the page, so it would be this axial position here. All right, so then where are they both going to go when they flip? Hopefully you can see that they will both move into this equatorial position. So the one on carbon 1 there, and the one on carbon 4 there. And so which one of these structures is going to be more stable, or which one of these conformations is going to be more stable, A or B? Hopefully we can all see that it will be conformation B here. Okay, I kind of stumbled over myself because I think I did the same example twice and I didn't mean to. So I was like, wait, what? But anyways, this is the name of the game here. So I'm going to put down a cyclohexane conformation or stereoisomer. Now, what would happen if, for example, okay, this is kind of this format that I see that Owl's using, where it's like a mix of two different types of representations here. But really what this is saying is that on this particular bond is actually an ethyl group, not just a methyl group. Okay, so if we have carbons 1 and 2, carbon 1, that bond is pointed out of the page. So I'm going to say that that's my axial bond here, pointed out of the page. And so that means on carbon 2, is this ethyl group in the equatorial position or the axial position? What do we think? I have this ethyl group right here on carbon 2. Is it going to be in the equatorial position or the axial position? What do we think? Give it a shot here. You got a 50-50 chance of getting it right. Okay, so this would be the equatorial position. All right, I think that that's harder to see than if it was a wedged bond that was pointed into the page. Then it'd be kind of easy to see that it would be the axial position. A dash bond pointed into the page, it'd be easy to see that it's the axial position. Because it's this wedged bond, you know, basically you have this choice. It's either going to be this one or this one. Well, which one is on the same side as our axial bond here that we said, right? So I know it's take a little bit of practice, but you want to convince yourself that because these two bonds are pointed in the same direction, I have to have this combination here of axial and equatorial. Because if it was this one down here, then they would be pointed in opposite directions, right? So since they're both wedges, since they're both pointed in the same direction, one has to be axial and one has to be equatorial. And so this is, I'm going to go kind of quick because we're running out of time. So this is a methyl group. And this one was our ethyl group. So then when we do our chair flip, this becomes equatorial, the methyl group does. And when we do our chair flip, this green one becomes the axial position. And again, that was our, our ethyl group. Okay, so then which two of these conformations is more stable? A or B? Cool. So because that ethyl group is so much bigger, it's going to sort of win. It's going to get its way, right? And it really doesn't want to be in that axial position. So that means it will be in the equatorial. It's going to win. Structure A will be the more stable conformation. The ethyl group will be in the equatorial position. So this one's the equatorial position, meaning that this is in the axial position. Okay, so again, this particular slide, I would sort of grab it and be able to kind of annotate. It's a pain in the butt to draw these chair structures on your own in your notes. I mean, you certainly can. But if you would download a page from that lecture slides and then sort of just annotate on top of it, kind of like I have here. And I think that that's kind of the easiest way to go about these problems. Okay. So we will finish up chapter 2 on Monday, talking about reactions of alkanes. We haven't gotten there, so that assignment's not going to be due until the end of next week. Yeah. And again, remember we have this quiz, and I kind of listed the topics here. All right, but other than that, I'm done with you guys. I will see you